Good morning. Hi, my name is Wenyi, Wenyi Wang. I'm a professor at MD Anderson Cancer Center, Bioinformatics and Biostatistics. So it is my great pleasure to be here today to teach again at CGSI. Last year I had fun. And so last year I talked about statistical methods development for tumor heterogeneity and evolution. So this year I'm going to switch gear and talk about the other long-standing research program in my laboratory on statistical methods development for cancer risk prediction. So we've been developing statistical risk prediction models and software tools for inherited cancer syndromes since 2003, 20 years ago. Actually 1 month before I started my PhD study. So I jumped on the project with my PhD advisor, Giovanni Parmigiani, working on perfecting and refining BRCA Pro for risk prediction. So for those who are not familiar with the concept, let's start with inherited cancer syndromes. I don't have a pointer, so I think I can just. So the top half of this graph, the illustration shows you a pedigree of history where the circles are the female, the squares are the male, horizontal line points to a couple, and the vertical points to the offsprings. All right. So and then you notice the individuals with a highlight. So these are the people who unfortunately inherited or developed a germline mutation in a particular gene, which then consequently led to a much higher chance of them developing a particular cancer type. So that's the kind of the background of inherited cancer syndrome. And then the same germline mutation are then inherited over the generations. So like I mentioned, I started working on inherited cancer syndromes with BRCA Pro for predicting germline mutations in BRCA1 and 2 for breast and ovarian cancer syndrome. And BRCA Pro is currently widely used in the clinic for genetic counseling for breast cancer patients. Then later on worked on, developed a MelaPro for CDKN2A for cutaneous melanoma, and then Panc Pro for predicting familial pancreatic cancer. Since I joined MD Anderson in 2010 as assistant professor, I focused my research interest in Li-Fraumeni syndrome, which is a fascinating syndrome. It's devastating but fascinating mathematically, which is mainly caused by a germline mutation in TP53. So coming back to this illustration, so these are the individuals who were affected with the mutation in TP53. What's difficult to characterize this syndrome is, as is shown here, that different family members with the same mutation may develop different kind of cancer at different age stage. So for example, early on in young kids, brain tumors, and then in adolescents, sarcoma, and then in young adults, breast cancer. And the same individual will continue to develop one cancer after another as long as they survive the first one. So it's devastating for these families, even though, and luckily, it happened rarely in the general population. But these families are extremely complex in the cancer phenotype that they present. All right, so before I jump into the risk prediction, I want to just give a little bit of background. Maybe you've talked to TP53 a lot in the CGSI, but why do we study TP53? It is a small protein with 393 amino acids. It was discovered in 1979, same age as me, by Sir David Lane and Arnie Levine. It is the most well-known, probably the most well-known, tumor suppressor gene ever studied and plays a central role to almost all cancer types. And therefore, TP53 gained the name of the guardian of the genome. It is called the guardian gene. So when the guard is out, so what happens to the genome? All kind of mess are much more easy to happen. So the elephant in the room here is the elephant itself. What do I mean by that? So elephant has 100 times more cells than human. So think of cancer. Cancer is the disease where cells turn to malignancy. However, people always wonder why elephants have a much less likelihood or probability of developing cancer. So finally, in the past 10 years or so, 10 years ago, researchers sequenced elephants' genome, and they discovered they carry at least 40 copies of TP53 as compared to 2 copies in the human. So it is reasonable for us to hypothesize that cancer risk in human genomes, with or without the guardian gene, is likely to be vastly different and therefore quantifiable. So let's first focus on the vastly different part. There comes Li-Fraumeni syndrome, which are mainly caused by, as I mentioned, by germline mutations in TP53. But let's go back in history a little bit. So Li-Fraumeni syndrome, it was introduced and defined by two pioneer population cancer geneticists, Fred Li and Joseph Fraumeni. They introduced the syndrome in 1969, 10 years before TP53 was discovered. And after 1979, TP53 discovered, it is not until 1990 David Malcolm published a paper in Science and then formally matched the two together and suggesting that Li-Fraumeni syndrome is mainly caused by germline mutation in TP53. So ever since that, 1990, now it's 2023, over the past 30 years, there's a huge development in LFS research and TP53 research, and they merge together. So nowadays, families affected with LFS attend research conferences together with scientists who study TP53, and they ask questions and they have conversation. So I had the opportunity and the fortune to attend such conference called LFSA conference every year and continue to be inspired by these families. Further on the drama side, LFS made it to Hollywood. So Grey's Anatomy, season 10, episode 14, You've Got to Hide Your Love Away. So Rory has a genetic condition that doesn't allow her to fight off cancer, the LFS. She advised her sister to not take a test to determine if she has the same condition because she said knowledge isn't always a good thing. And this was aired in 2013, and I have to agree that the writers probably need to be paid more so they will have time to do literature search more carefully because 2011, this seminal paper came out of David Malcolm's lab in Toronto showing that the biochemical imaging surveillance, mostly MRI, which are currently being implemented in most major hospitals, can substantially improve the survival outcomes with families with LFS. So early detection is essential for these families. So knowledge is important. So with that, the available screening protocols, now the researchers are more, or clinicians are much more motivated to identify which families are actually LFS. Okay, so what's the challenge? So let's look at, by now you must be very familiar with this pedigree, circles, squares, offsprings. The challenge here is that there are so many different types of cancers that are presented in these families, which then confuse the clinicians with other cancer syndromes. So this was the very first, we call it classic criteria that were developed for LFS are simply 3 lines of criteria. So if that fits, then we suspect that this family is LFS. But the problem is it is really not that sensitive, and it is too stringent. Later on, the French group developed a Champray criteria, which is still currently being utilized in the clinic at genetic counseling sessions. So which is basically a much longer list of criteria that this family has to hit, and the genetic counselors, they kind of remember all of the cutoffs and then be able to check Champray or not. So higher sensitivity, but now this time is low specificity. But again, these are all written out descriptive criteria. So our goal is to develop quantitative risk prediction tools that are of high resolution and provide more precise and accurate prediction. So how do we do that? So probability assessment tools for risk counseling. Let's first think of the key probabilities that we're trying to generate. There are 2 of them. First is the probability of an individual carrying cancer-causing inherited DNA variant given family history. So we call it mutation prediction. Second is the probability of an individual developing cancer in future, if unaffected at the moment given family history. So it's a future cancer prediction. So how do we derive these 2 probabilities? there are two types of models, main categories of models. Empirical models, which are like regression, logistic linear regression, with covariates being the summary statistics of a family history. For example, the total number of cancers occurred, and then the average age of diagnosis, something like that. But it has to be summary statistics to be able to feed into a regression model. The other category is Mendelian models, which is what we've been developing. So the concept of Mendelian model is explicitly model the Mendelian inheritance within the pedigree over the generations so that we can account for everything that we're seeing here, the type of cancer, the relationship of family members, and the ages that has been reported. All right, so with the Mendelian models, using the Mendelian model framework, we developed LFS Pro. First, the three key aspects of LFS Pro. First is key assumption. So there are no other forms of familial aggregation other than the genetics that are transmitted in the family. The second is we account for probability of de novo TP53 mutations. Has de novo, does that word has come up in the CGSI yet? De novo? So just new, new germline mutations, good. All right, so, and then the input data is simply a data matrix with rows being each family members and the columns showing the relationship of each individual to the counselee through individual ID and parent ID, and their cancer status and their age of onset if affected or age at last contact if unaffected. Notice that we do not take the mutation, the genetic testing information into account in the input. And then the output are the two probabilities that I mentioned, the mutation probability and then the cancer risk probability. All right, so here's an illustration to help you understand what exactly is, what's the utility of Mendelian models. So let's start with panel A. The arrow points to the counselee who had a sarcoma at age 12. She has a maternal aunt, had breast cancer at 40, and a sibling of the maternal grandmother, sarcoma at 25, and then the great-grandmother, leukemia at 27. So we put this into LFS Pro, probability 57%. So notice that, let's focus on the maternal grandmother. So she is unaffected at age 42, which slightly argue against our germline mutation that are being transmitted in this lineage. So how about we switch the grandmother with her sister? And now the grandmother had sarcoma at age 25. Notice that these two at the summary statistics level has no difference. Okay, same amount of cancer and the same age of onset. The relationship switched, and then we give a much higher probability for the counseling. All right, so C is simply adding another cancer in the family, easy, even higher probability. Okay, so with D here, we're testing out how about we say all of the founder in the family, the great-grandparents, stayed healthy at a much later age, which then argue against, at this stage, having a germline mutation in TP53. So either there's a de novo event that happened here, or there just shouldn't be a mutation. So therefore, LFS Pro gives 60% if de novo is allowed, and 2% if not allowed. All right? So, in summary, so Mendelian models provide risk assessment in a finer resolution and response to detailed family history information. Any question here? I heard some. Yeah.
Sorry. So is this the probability of getting cancer, the probability of having a mutation?
Mutation so far. Yes, I'll talk about cancer later. Yeah. All right. Okay. So digging deeper, so there will be more equations coming up. Just brace yourself. All right. Okay, so how do we do the calculation? All right. Okay, this is carrier probability of ICE individual with relatives. Am I in the camera? Is that? Okay, all right, doesn't matter. So how do we calculate? So our desired probabilities, probability of GI given age, all right, genotype of individual I. So the calculation is based on first an updating step that we're using family history, probability of age given GI, to update the probability of GI. When age is out, this is a population level estimate, or and can be easily derived from allele frequency. So the key is how do we calculate pH of given GI? So here, when we condition on the genotype, all of the genotype in the family relatives, the minus I means all the other relatives other than individual I, then we can further separate out this equation into two components. One component, based on conditional independence, one component is the product of all family relatives, probability of DJ given GJ, genotype, and then disease status. And the second is the probability of that specific genotype configuration, the chance of that occurring given the GI. All right? So just remember orange or red, blue, and green, 3 parts. All right? So red and blue are usually provided and studied specifically by genetic epidemiologists. You can find papers that focus on providing these two estimates. And then green is a computationally intensive part, which I'll talk about a little bit in the next slide. So one of a particular kind of simplest dumbed-down case of P of G minus I given GI is listed on top. So probability of a trio family, genotype of the mother equals to big A, GF of big A small a, and given G child, big A. All right? So how do we calculate that? Even with a trio, it's not that straightforward. Go ahead.
I'm so sorry. I think I'm a little confused. So you have the genomes of all these people?
Not genome. This is one gene. It has nothing to do with genome. This is genetics, not genome. Yeah.
You don't have the full genome of the family members?
We're just focused on TP53 one gene. We don't need the genome.
But you have the information if the people in the family has it or not?
No. This has nothing to do with genome. Or
But the gene, like the specific gene you're looking at, do you know if the family members has it or not?
oh no, we don't. We don't even know. We don't know anyone's genotype.
Okay. So
This is all probability. It's like
so
you know.
you only have their cancer status.
Exactly. Yes. There's somebody wrote. Yeah. Oh.
I just want to say, you might want to repeat the questions. Can't hear the question.
Oh, yes. So the question is, do we know the genotype of any of the family members? And the answer is no. Yeah. So all of them are unknown. So hence we wanted to calculate the probability. So this is just so we have 3 genotype per individual. So with a trio, we have 3 to the power of 3 possible genotype combinations. Yeah. And then with this, with the green part here, we have 3 to the power of 2 given one particular GI. Right? All right. So with that particular the one probability, what we do is simply with trio, we can still kind of list out this, I call it hash table. So on the top, we have probability of GC equals to big A, right, given the father's genotype and mother's genotype, which is a 3x3 table. And similarly, big A, small a for the GC and small a for the GC. So we need to look up particular values in each of these hash tables and then to be able to back calculate the probability on the top. You can take a picture and then do the calculation, and we can discuss the result. But yeah, it should take you like a few minutes. All right, so the point is, in a genetic counseling session, the common size, family size is 20, between 20, sometimes 30 and 40. So you can do the mental calculation of how do we actually get all of these joint genotype probabilities. So that's why I said computationally intensive. Luckily, this problem has been solved by giants of the field. Peeling algorithm, 1971, Alston Stewart. They've already thought about this and came up with a very efficient algorithm that peels through. Anyway, I can talk offline about this. So we simply took the peeling algorithm and implemented inside of AlphaFuzz Pro to be able to resolve the computational intensiveness of the green part. But the mathematics is actually straightforward. All right, so I'll hold off on describing the blue part. The rest of the lecture will be about how to improve on the blue component. But for now, this is our LFS Pro validation using the previous published estimates of the red and the blue, all right, which are very rough, which we will refine later. So here we have the opportunity to obtain a total of 867 families from 3 major studies, 2 in the US and 1 in Australia, with 1,300 tested individuals, positive and negative. So I'm showing you the result with ROC, receiver operating characteristic curve, with the curve closer to the upper left corner being a better performance in discriminating a 0 and a 1 outcome. So we did very well with MD Anderson cohort and an NCI cohort. Remember, these are also independent, that none of these data were used in the training. However, we didn't do as well with the International Sarcoma Kinship study, which was performed in Australia, and were focused solely on adults. And then we suspect most of the reason is that the underlying TP53 mutations carried in these patients are quite different from the other 2 LFS cohorts in the US. And I'll come back to it at the end of this lecture. All right, so moving up beyond LFS Pro. So how do we establish a Mendelian model? There's 6 major steps. So we identify cancer of interest with known genetic effect, and we characterize the effect of the susceptibility genes in terms of prevalence, the red part, and the penetrance, the blue part. And we set up the model based on Mendelian transmission, obtain independent data, and evaluate model performance. And we make user-friendly software and tools and then disseminate. And then we continue to update the model with additional genetic information in order to improve the model performance. So next, I'll focus on updating the second part, which is to further improve the penetrance estimation. So penetrance is an important parameter for genetic epidemiology and also for our models. Its definition is the probability of the primary cancer onset up to by a certain point, and then conditional on the susceptibility gene mutation status. So it can be for non-carriers, which will be much lower, and then for for mutation carriers. And then on the top, reminding you of the equations that we use for risk prediction. So we're focusing on the blue component. Previously, in the literature, the blue component for LFS Pro was combining all cancer types together as just simply having cancer or not, which is not at all capturing the complexity presented in LFS, as I'm showing you here in one of our studies, meticulously collected over 40 years of time by my collaborator, Louise at MD Anderson. So here we're showing you the number of cancer patients who had a mutation, were tested that a mutation, carrying mutation or not carrying a mutation, and all the family relatives that were not tested. And we were interested in characterizing different cancer types and the onset of the first primary and then the second primary cancer separately. Well, what you can see here is the sample size drop substantially when we try to characterize more specific phenotype within this family, as compared to the relatives. These are also cancer patients, and their relatives, meaning that they are likely to carry also a mutation, except that they were never tested, like what you asked. Yeah. So our whole kind of past 10 years of effort was to develop integrative models that we can include cancer patients in the family members here of the tested individuals to maximize the sample size to improve the penetrance estimation of the specific types. So how do we incorporate it? We add probabilities. We first calculate every individual's probability of carrying a mutation and use them as weights to then include their cancer outcomes, age of diagnosis, into the penetrance calculation. So that's basically the gist of my talk. All right. So now the math. Again, the overview of penetrance characterization. Since we're looking at families, when we modeled the data, the first thing we consider is whether there's other non-genetic component that introduce family-specific variations, and we therefore introduce a frailty. And then with inferiority incorporated, we calculate a family-wise likelihood, assuming that each family are independent, but within family, family members are not independent. And then we account for ascertainment bias. So by the way, each of step, the reason I highlight them is each of them have a whole field researching and then trying to perfect that particular step. But we're basically taking all of the state-of-art approach, combine them, and then focus on this step, which is what we introduce as the new component. Okay? Ascertainment bias is important. Why? Because we're ascertaining a very rare disease. And we can't, without correcting for this enrichment of a rare disease, then we will be estimating a very high probability of a general population developing cancer, which would not be right. Bayes estimation is Markov chain Monte Carlo. And then after the MCMC, we estimate the parameters, and then we set things up for predicting cancer risk. All right, more equations. Let's see. So G, genotype, and H correspond to phenotype, where we have Y, which is the observed survival time, and then T is the time to the case type of cancer, as compared, whereas the rest of the cancers didn't happen. And then D correspond to the disease status or cancer status, yes or no. And X correspond to the baseline covariates, such as gender. Some subscripts: family index I, subject index J, and cancer subtypes index K. All right. So remember, K is most important. Remember K. All right. So first of all, we wanted to study cancer-specific age-at-onset penetrance. Here we introduce the cancer-specific age-at-onset penetrance, QK, which is defined by, as I mentioned, probability of an individual developing cancer K by age T when none of the other cancers happened. All right. So there's a competing risk component in here. All right. So the key point is that we want QK, but QK needs to be derived from lambda K. So this is like survival analysis 101. Lambda is the hazard. Probably you all have heard of Cox hazard, proportional hazard Cox regression model. Cox PH, anyone? Yeah. Okay. Great. So the proportional hazard model is modeling the hazard, and and this equation shows the relationship between hazard and our QK, the desired probability or the penetrance parameter. Okay, so what I'm saying is that just capture the key point because of the time limit. All right, so moving from QK to lambda K relationship, and then now we move down to this equation where we have lambda K is modeled by a baseline hazard and an exponential term, where beta K are the coefficient, and Z is, Z is basically the combination of a genotype and other baseline covariates and their interaction term. So that's the kind of the basic setup of our model. All right. So once we set up the hazard model, we can calculate the likelihood for each individual based on partial likelihood, and then take the product across all family members. Notice that this term, we go back to the green component. All right. The key here is that every time we have to specify every family member's genotype as known. So it's conditional on a specific configuration of the genotype. And that itself is a probability which we will calculate using peeling algorithm. And this is how we stitch together all family members by assuming that when the genotype is known, then they have independence, therefore the product. right? And then we simply sum over all possible genotype configurations. We have peeling algorithm. We're good. It's computationally, it's taken care of. All right. So then, this gives us a family-based likelihood. And then we take the product across families to get the full likelihood. Okay. All right. So I'm going to skip the MCMC and now show you some results. All right. So this is done by my former postdoc, Songjun Xin. So we applied our model to estimate the competing risk outcomes of breast, 66, so these are sample sizes, sarcoma 211, and all other cancers combined 228. The reason we do that is because breast and sarcoma are the two most common cancer types in our MD Anderson cohort. And here is the estimated outcome, our Q case on the y-axis, and the ages on the x-axis. What we saw is, first of all, sarcoma overall, it kind of levels off when the person gets older, but it does have the highest hump at the very beginning, as we would expect. That sarcoma happens mostly in adolescence, and that the breast cancer catches up quickly and is the most present the highest risk in this family. What's most interesting, though, is we then took this penetrance curve based on TP53 with BRCA1, 2 based penetrance. And this is a zoom-in view. The dotted lines are BRCA1 and 2 based penetrance. All right? And the solid line is TP53. So ignore this part. This is just smoothing. It's a kind of artifact. But between age 20 and 25, we see a much, much stronger signal, a probability of TP53 mutation carriers developing breast cancer than BRCA1 and 2. All right, what does that mean clinically? It means that when clinician sees a patient, a breast cancer patient, developing early onset breast, they should really strongly advise the patient to also consider TP53 testing as compared to BRCA1, 2. And that TP53 mutation carriers are super sensitive to radiotherapy, so that these patients should not be sent for radiotherapy, which will then cause additional cancers.
What is WT and MT?
Sorry, say.
WT and
Oh, wild type,
What
sorry.
is MT?
MT mutation. Yeah. Yes?
Cancer-onset time, and do you refer to, do you use the time of diagnosis, or do you do like some kind of interpolation because cancer starts before diagnosis?
Sorry, say again. Oh, yeah. No, this is, we're talking about clinical diagnosis time. Yes. This is not about evolution or, yeah, so it's a different concept. But I'll show you more on the clinical side, which can help you as well.
Or not simulated?
These are real data.
So you do have the genotype.
Uh,
If you have wild type and mutant, you know they're.
oh, yes. These are, yes. But for, so this is for characterizing penetrances. And then we will rely on the genotype of whoever were tested, but then we also want to include everybody else who were not tested to boost up our sample size. But when it comes to risk prediction and risk counseling in the clinic, we have to mimic the clinical scenario, and that's where no genotype information should be given as input. But that's for risk prediction. This is for penetrance estimation. Penetrance estimation is the blue component, which will then go into the risk prediction. Yes.
So then the blue component,
Yes.
that would mean that you're censoring people who are below the age of like the common incidence before it starts moving up.
So you mean the utility?
Someone is let's say 20 and they have the injury count that they have breast cancer, then they won't be like control because you haven't observed them long enough to know if they actually will have breast cancer.
Good question. So the question is whether.
So you use the cumulative incidence curves to weight people. So they will definitely be a control if they are like I don't know, the 80 and it's flattened out. Because then you see if they don't have cancer in their 80, they're not going to be having it for this specific number.
All right. So we can talk offline about this.
Okay.
Yeah, so I think there's more about this PDF, like a probability distribution function, cumulative distribution function, and then how is it interpreted in competing risk outcome. But we can discuss offline. Yeah.
So what if a patient does have TP53 mutation and BRAF 1 or 2 mutation?
Well, yeah, I don't think we will see them because they probably die pretty early on. But the more concern is that in the inherited breast cancer syndrome, they are definitely finding patients who are actually carrying TP53 mutations. And like I said, these patients should be treated differently. For example, certain like radiation therapy are extremely detrimental to TP53 mutation carriers because their genome are very fragile already. And by the way, this finding was confirmed recently by a British breast cancer study. So I just heard a talk at LFS about this. All right, switching gear again. So now we wanted to model multiple primaries. Just a quick note. So we started out looking at this problem we were scratching our head. Now how do we approach this? So we decided to kind of dissect out the problems. So first we dissect out characterizing competing cancer outcomes within the first primary. This is the second dissection. So here we're ignoring different cancer types. We just focus on studying the repeated occurrence of first primary and the second primary within the same individual. And the other reason we wanted to do this is because there are also more available statistical models we can implement. So here, the key parameter to remember is the gap time, WJ. All right, J is the J's disease. So the gap time just simply means from the birth to the first cancer, that's gap time 1, W1, and then from the first cancer to the second cancer, that difference, right? It's not the age anymore, but that difference is the second gap time. So what I'm showing you is that we first check the second gap time versus first gap time. There's a relationship, there's a correlation, so they're not independent. And then second, the actual distribution of gap time. Blue is the second. I think. So the solid line is the second, and then the dashed line is the first. So they're also not identical. So when the gap times are not IID, what do? We use non-homogeneous Poisson process, and the non-homogeneous correspond to the non-IID component. All right. So with implementing non-homogeneous Poisson process on our data, and so here's the result. So here, what I'm showing you again is the Q, right? The penetrance estimate across ages. So we say time because when this is actually gap time, so it's not the absolute age. But here, in both male and female, dotted line and solid line, we saw that when the first diagnosis happened early, so 0 to 20, versus mediate 21 to 40, or late 41 to 60, the second penetrance curve changed. The first cancer happened early, then there's a long wait time for the second to happen. But if the first happened in a middle-aged person, then the second cancer will happen almost right away. And this, again, has clinical implication on how these families can be tracked. And again, we found, when I talked to LFS families, they were confirming that what we have found mathematically is indeed happening in their families. All right. Yes?
Extend this model to what as gap time, and can you also think to use like longitudinal models, especially for the people in the early and late onset, since the gap time is shorter?
So the question is, can we use other longitudinal models to model this? The answer is probably yes, but we also had something in mind in kind of future development, which I'll show you in the next model. Yes. Okay, so reminding you, overview of penetrance estimate characterization, just all the work we did to get the blue part right. So frailty, family-wise likelihood, ascertainment bias correction, MCMC, running MCMC, it takes weeks to finish estimating the parameters and setting up cancer risk prediction. All right, so now this is a graph representation of what we did. Look at T1, the first primary, competing cancer outcomes, or ignoring the cancer differences, characterizing T1, T2, etc. All right. So once we've done these, we were like, okay, we're ready to tackle the real challenge, which is to characterize competing cancer outcomes in T1, T2, and T3 all together in one go. And we found that the non-homogeneous Poisson process, to answer your question, is a nice framework for us to further expand. All right. And as we develop this, we realized one thing is our study is motivated by LFS. But what this model can do is beyond this rare syndrome, as in there will be more than 20 million cancer survivors living in the US by 2026. And most of the clinical management of cancer survivors are not personalized or fine-tuned to their status as being surviving a cancer already. So clinicians need help to characterize the development of these cancer survivors and to be able to develop fine-tuned management plans. All right. So again, math, but just focus on this, WKL. All right. So previously we have WL, which is gap time. Now we add a subscript K because here we account for competing cancer outcomes. So I'll just skip the further math. Again, relationship reminding you, now we have QKL, which is our penetrance, which can be calculated using its relationship with the lambda K, where lambda K we've characterized using proportional hazard model to incorporate the genotype and other baseline covariates. All right, so the same gist. But mathematically, we also found that introducing competing risk to a recurrent event model has not been done before. We looked and looked in literature. Only one group had made an attempt, but I think our model can do much better. But I can't help wondering why is it not developed so far, because it's such an important problem for studying, for example, cancer survivors. Fast forward a year later, we have our model, fully round MCMC parameters all estimated. Now we perform a validation in an independent cohort. This is another MD Anderson cohort that was not used in penetrance estimation. So on the left, I'm showing you the validation in terms of our joint model's ability to take care of the competing cancer outcomes within the first onset cancer. Looks good. And the bottom is joint model's ability to take care of the onset of the first and second primary without considering competing cancer types. Looks good. And here is our ultimate goal, which is to predict competing cancer outcomes in the second primary. Not as good as here, but still reasonable. Thumb up. So, and this is a comparison. This is we call it naive model, which is simply to assume independence and IID, identical independence distribution, to multiply our competing cancer outcome penetrance for the first primary with our recurrent event outcome for the second primary. But that does not work in predicting in the real data. All right, any questions here? This is all real data validation. Oh, okay. So here's to show you another result. So we thought, we looked at the cancer survivor registry and found that single primary lung cancer is one of the most prevalent cancer types. And screening for lung cancer has been going on forever with millions of dollars invested. But when we looked at the criteria for people who qualify for screening, they have not specified a cancer status, whether this person have developed a previous cancer. And we were worried about it because there are recent literature specifically about breast cancer patients developing lung cancer as the second primary, which is expected because of the exposure of the treatment in the same field. So we took, inside our LFS data, we took those individuals who actually developed lung cancer. We indeed have some, and then make our prediction, and we found a very good performance. So this is also reassuring and encouraging, as in how our model can be useful for the screening practices for for cancer survivors, which we believe is coming, up and coming, and need a lot of work in it. Switching gear to clinical dissemination. I have shown you a lot of validation. These are validation based on cohorts that were meticulously collected over the past 40 or 20 years. So what about clinically collected data? So here we're collaborating with Banu Arun from MD Anderson. She chairs the clinical cancer genetics program. I'm showing you this comparison. The green is the clinically ascertained, I call it clinical counseling-based data. And then the red is the research protocol-based data. So when we look at the family members, there's not much difference in the ballpark, but we do observe this really high tail in the research protocol-based cohort, as we have years to follow them up. So there's certainly some families, them being diligent, we are able to collect more and more information, and with a much larger family pedigree. And on the right, I'm showing you the missingness. When age-at diagnosis or last contact is missing, then risk prediction tend to be less accurate. So on the top is the clinical counseling-based data, CCB, that has much higher missing rate as compared to, at the bottom, the research-based cohort. All right. And it's kind of afterthought. So I talked to the genetic counselors. So you can see that in terms of data collection, there's a huge difference comparing the top 20 years of iterative annual process, sending Christmas cards, filling out the surveys, as compared to the bottom clinical counseling, 1 hour session, and was at a maximum 30 minutes dedicated to collecting family information. All right, so this is CCB and that's RPB. So there's a huge difference in the data collection. Now, with CCB, can we do well? This is the critical question why research prediction models, so many were developed, so few made to the clinic. The answer is reassuringly yes, but less good. So here are the area under the ROC curves that our AUCs are good using our updated model for the cancer-specific outcome penetrance or the multiple primary cancer outcome penetrances. But the 0.75 is still 0.1 lower than the 0.85 we were able to achieve using LFS Pro on RPB data. All right? But interestingly, even the clinical criteria developed by clinicians, they also dropped by 50% in terms of sensitivity when applied to the CCB data. So that's a little bit surprising. All right, dissemination. So again, collaborating with Banu Arun, and these are the two leading genetic counselors, Jacinda and Jessie. So we worked out this Shiny app, not just the Shiny app here, for all of you, most of you, Shiny app, easy to develop. But the point is, we managed to get MD Anderson EPIC system. The genetic counselors will click a button, say suspect TP53, and the EPIC system will export data frame in the exact format that LFS Pro needs. Therefore, the genetic counselors can just upload those data to this Shiny app and then roll out LFS Pro and then be able to look at these the predicted values and the pedigree. Right? So we got that working, and that gave us then the first-hand knowledge on what a risk prediction model like LFS Pro can actually contribute in real genetic counseling. This is a prospective study that started in December. Yeah, I'm almost done. Yes, in December 2021. So here on the left, LFS Pro, on the right, clinical criteria, Champray. We still make mistakes. There's so many weird families, family structure, but overall, we are already doing better. Focus on the red, we're doing better than the clinical criteria. And GC counselors, they filled out survey after seeing the patient, and 84% of them say they will use LFS Pro. And I'll skip what their comments are. Some are encouraging, some are informative. Banu Arun initiated the patient participation survey. So far, our consent rate is low, but we think this is because this is a software tool that we're asking patients whether they're willing to participate for a clinical trial. So this again is informative and helpful for us to devise appropriate responses to improve. All right, finally, to conclude, so we have developed advanced statistical models based on risk prediction, which are developed and validated using clinically collected data sets. Risk prediction models trained by research cohorts can be less accurate in clinical cohorts, but are still useful. And quantitative risk estimates add another layer of nuances to clinical criteria for effective communication with patient families, and collecting survey on GC and clinicians are useful to refine and pinpoint the utility of risk prediction. I want my tool to be used in the clinic, so that's the goal. On the other hand, to closing with a teaser, so there's still a lot of interesting questions to be addressed with TP53. So for example, grouping TP53 mutations in terms of their distinctive functional outcomes. So my postdoc Xiaoqian Liu are recently developing a penalized Cox regression with a new penalty called distance penalty. And this is a preliminary result that we're able to identify TP53s in distinct outcomes. And with that, I'll end and thank you for your attention.